In the name of God, today we continue our lectures with another definition with the name of amensalism. Amensalism. If you didn't remember, last lecture we were talking regarding competition. In competition, we told you that when two populations have the same resource, when two populations have the same food, and it is limited food or in limited space, they compete to have it without attack each other, without production any toxin. So it was competition. Today we focus on amensalism. In amensalism, it is a type of competition that one population can produce toxin to inhibit or kill the other population and receive food without problem. So what is the difference between competition and amensalism? In competition, what is the similarity? Similarity is that in competition and amensalism, two populations need the same resource, the same food, and food is limited. It is similarity. What is the difference? In competition, winner is the population which can absorb, can receive food faster, grow faster, multiply faster. But in amensalism, the winner is the population which produce toxin and kill or inhibit the other population. The other name of amensalism is antagonism. Antagonism. So
You are currently the only person in this conference.
Now is it okay? Can you receive my voice? Is it okay? Oh, very good. So I repeat, I don't know from which point you didn't have voice, so I repeat. So during amensalism, one population produced toxin. This population is resistant to its toxin, but most the other populations are sensitive. Sometimes we use antibiosis or allelopathy. The same definitions for amensalism. The same definitions for amensalism. For example, some populations can produce virucidal factors. What is the meaning of virucidal? Means virus killing factors. Why one population kill one virus? Is it any food? Is it any common food between them? Yes. One population wants whatever they have. What do they have? They have many substrates, many sugars, many proteins in their bodies, and virus wants them. So one population wants to defend and wants to use whatever they have, and virus wants to use whatever the population have. So some populations can produce some toxins or some virucidal factors and stop or kill the pathogenic virus for them. For example, some populations can produce fungostasis or fungostatic factors. Suppose there are some common resources between one population and fungi, or even though one fungus wants to use whatever one population have as a parasite or as a predator. So that population as parasite, not predator, as a parasite. So some population can defend regarding whatever they have, regarding those food they have. They don't want give their foods to fungi. So they produce some toxins. And when toxin is inhibiting the pathogenic fungi, we call it fungostatic factors. Fungostatic factors. So accordingly, once an organism establishes itself within a habitat, it may prevent other populations from surviving in that habitat. Once an organism establishes itself within a habitat, it may prevent. How preventing? By production toxins. Other populations from surviving in that habitat. Example number 1. Suppose some microbes are living in the domain of ruminants. You know, some animals like cow contain some microbes in their rumen. Some populations are anaerobic heterotrophic microbial populations. Some are anaerobic heterotrophic bacteria, which can produce volatile fatty acids. They can produce volatile fatty acids and excrete it in the rumen. So, most coliforms like E. coli cannot grow there. You know, in all of us, in our alimentary canal is full of coliforms, and especially E. coli is the dominant bacteria in our intestine. But some present, some native bacteria in the rumen of ruminant don't want these competitors to be there, so produce toxin. What is their toxin? Their toxins are volatile fatty acids. Or for example, there are some native microbes on our skin. There are some native bacteria on our skin, and they don't like some other populations to be present there. So some of normal flora on our skin can produce a toxic substances with the name of fatty acids. And by production of fatty acids, most the other populations cannot be present there. So they win in competition by production of toxin and inhibit or kill the other populations. Example 3. For example, in females, at the end of urinary tract system, urogenital system, at the end part, some microbes are present and they can produce acid because they don't want the other competitors to be there. And one important competitor albicans, and it is causative agent for candidiasis. So in normal case, normal flora vagina produce acid inhibiting candida albicans. What is this? This is amensalism. It is antibiosis or allelopathy. Example number 4. Consumption or production of oxygen. What is that? Okay, so sometimes, in some cases, you feel oxygen is very good because we as a human, we are aerobic. Synthetic bacteria like cyanobacteria are present in one place during photosynthesis and carbon fixation and sugar production. They also produce oxygen, which oxygen kills all obligate anaerobes. It kills obligate anaerobes. Okay. So oxygen is produced by some populations is amensalism, is antibiosis for obligate anaerobes, and in opposite directions. Sometimes consuming oxygen, for example, when one facultative anaerobe is in one place, use all of oxygen present in that habitat. Absence of oxygen is toxic for obligate aerobes. So if oxygen is used and finished, now that habitat is toxic for obligate aerobes. So consumption is toxic for some microbes, and production of oxygen is toxic for the other microbes. For example, algae produce oxygen, so it precludes growth of obligate anaerobes. Example number 5. Some populations by production of ammonium can kill some the other populations. Production of ammonium by population 1 is deleterious for population 2. Population 1 produce ammonia during the composition of proteins and amino acids. Okay, and by help of this toxic production, population 2, like nitrobacter, which they are nitrate oxidizing population, is inhibited or killed. Example number 6. For example, one population like yeasts, they can produce alcohol or ethanol. So whenever you go to the hospital, you smell alcohol, because mostly alcohol is used for disinfection. Disinfection, different things, especially before injection, syringe injection to one person, first disinfecting the skin, or before blood transfusion or whatever, or serum injection, first using alcohol for disinfecting the skin. So very easily we know alcohol can kill many microbes. And during the corona days, most of people have a small bottle of alcohol or ethanol, and when going somewhere, disinfecting hands and whatever they have. So population 1, like yeast, produce, excuse me, 1 minute, again I cut it. Excuse me, again we can continue. Because for a long time during the day we teach, so sometimes speaking and teaching is difficult. It is needed a few a little water to drink. Okay, so very easily we know that ethanol, which is, thank you very much, is produced by some microbes, can easily kill most other populations. But it is not so that all populations to be killed. Some populations can have defensive behavior. For example, in some cases, when one yeast produces alcohol, some bacteria, such as acetobacter, can convert alcohol to acid and neutralize it, neutralize acidic situation, like production of acetic acid. For example, in vinegar production. So how vinegar is produced? Acetobacters receive ethanol and change it to acetic acid. So it is vinegar. So this bacteria can be resistant against alcohol production by yeasts. It is done when oxygen is available. Acetic acid is also inhibitory to other bacteria. Acetic acid is also inhibitory to other bacteria. So the resistant bacteria to alcohol now by production of acid can kill or inhibit many other populations. What are we doing? We are teaching, we are explaining about amensalism. It is a situation which one population can produce toxin to kill or inhibit another populations to receive nutrients and whatever they need as a natural resource. Such inhibitory compound in food, lactic acid, propionic acid in cheese. Don't forget that in ancient time, in old time, people could have milk, receive milk from their domestic animals like cows, camels, and sheep and the others. But milk was very fast spoiled. So people in the ancient time could change milk to yogurt, change milk to cheese, and can have it for a longer time. But by help of work of some microbes, and these microbes by changing milk to cheese produce lactic acid, propionic acid, which they interrupt, they inhibit, they kill the other populations which could spoil in milk. Acetic acid in vinegar is also a natural preservative. So bacteria produce it and inhibit growth of spoilage, causing microbial populations in such a products. Inhibitory substances produced by microorganisms in food industry may also act as preservatives. What are preservatives? Those things which we can use in food and making food remaining for longer time and avoiding the spoilage causing microbial populations growing and affecting food. One of the most important example regarding to the antibiotics is the production of antibiotics. Production of antibiotics. So some bacteria, some microbes, some different types of microbes have the ability to produce antibiotic. For what? To inhibit or killing the competitors during amensalism or allelopathy. In this case, population 1 produce antibiotic that in low concentrations kill or inhibit growth other populations. But we can or we could indirectly using the amensalism of antibiotic producing bacteria. How can we do it? We can find those microbes which can produce antibiotic. So when we have disease, when we have infectious disease, and we can receive antibiotics from some types of microbes and use it against pathogenic microorganisms. It is using indirectly amensalism. But we shouldn't forget that some bacteria can be resistant to some antibiotics in different ways. For example, some populations are capable of degrading antibiotics, or they have another strategies not allowing antibiotics come inside the cell, changing their receptors. So by many different ways, some microbes are now resistant to many antibiotics. And we have multiple drug resistant bacteria. MDR, multiple drug resistant bacteria. Antibiotic production is important within microenvironment and certain conditions. In high organic matter, opportunistic microorganisms like zymogenous populations grow and produce antibiotics. Antibiotic production may aid in initial colonization of a substrate, after which antibiotic production may no longer be required. So sometimes antibiotic production is inducible. It is induced by environment. When competitors are there, those populations produce antibiotic to inhibit or kill them. But when they are inhibited or they are killed, so antibiotic production is reduced. For example, when there are some terrorists, policemen are shooting. When all of them are arrested or killed, there is no need to continue shooting. Yes. So microbes in the environment, if competitors are there, produce antibiotics till the time it is needed. When competitors are less or there are not any competitors, maybe those antibiotic producers stop antibiotic production. One example in this regard we can tell you is Cephalosporium gramineum. Cephalosporium gramineum. As its name shows, it is a fungus, and gramineum says it is related to wheat, but not a wheat, not a live wheat in farm. This fungus attack to wheat after harvesting. When the wheat is in the shop, when the wheat is in the store, it goes to use it as food, but it is not alone. Some another fungi are there, and they are competitor for this population. So when Cephalosporium gramineum, that is a pathogen of wheat and growing on dead wheat tissue, then this population produce and secrete antifungal substances. Why antifungal substances? Because other fungi are competitors. Other fungi are competitor for this fungi. So this fungus by antifungal substances exclude, stop, inhibit, kill the other populations and can use wheat alone as their food. But some strains of Cephalosporium gramineum lack the production of antifungal substances. So these strains are there, the other fungi are there, but it is not so powerful. So the others are winner and Cephalosporium is loser when cannot produce antibiotic. But this strain is winner because it is equipped with antifungal substances production. Okay? Another example is the presence of some microbes on the skin of one animal with the name of hog. Trichophyton mentagrophytes is a fungus which is present in the skin of this animal. Trichophyton mentagrophytes. And this fungus can produce penicillin like penicillium. Can produce the antibiotic penicillin. So when penicillin is there, this antibiotic kill all bacteria, all sensitive bacteria to penicillin. So when this fungus is present on the skin of hog, produce penicillin, and penicillin kills all bacteria which they are sensitive to penicillin. But some bacteria are resistant to penicillin. So penicillin resistant bacteria find this fungus as a good friend. Penicillin-resistant bacteria are the followers, are the satellite for this fungus. Why? Because they remove their enemies indirectly. Penicillin-resistant bacteria doesn't have the power of amensalism. They don't have the power of toxin production, but indirectly use the amensalism activity of a good friend with the name of Trichophyton mentagrophytes. So, Trichophyton mentagrophytes allows Staphylococcus to enter into the amensal relationship with non-penicillin resistant population that attempt to colonize the skin of hog. Penicillin resistant Staphylococcus has an adaptation to neutralize a mensal influence of Trichophyton. Adaptation here could reflect coevolution or coexisting microbial populations. Okay, another item, another definition is parasitism. Parasitism. Parasite. What is parasite? See, in competition, no attack, no toxin. In amensalism, toxin production. In parasitism, attack. There is attack of parasite to host. Attack of parasite to the host. Normally, parasite is smaller than host. Normally, parasite is smaller than host. Normally, the duration of contact between parasite and host is long time. Why? Because host give food to the parasite, and parasite don't want to kill food producer. So the contact between parasite and host is longer, and size of parasite is mostly smaller than the host. What is the parasite? Parasite is a living organism, live in or on the body of a host, receiving food, benefit, and harmful for host. See, there are a large number of microbes present in our body. There are a large number of microbes present on or in our body. They are there to receive food, they grow, they multiply, but they are not harmful. So they are not parasite. So right now we don't have any parasite. And we don't feel bad. We don't feel any disease. We don't have anything to tell the physician. But if one pathogen come to my body, if one parasite come to my body, now it is harmful. If it is not harmful, previously we explained, it is commensal. So right now, many commensals are present on and in our body. And sometimes during the year, or sometimes during many years, some pathogens, some parasites can come in or on our body and make us problem. And we should go to the physician and get antibiotic or specific drugs. Okay, so parasites benefit derives nutritional requirements from harmed host. Relatively long period of contact, direct physical or metabolic, is there. Usually parasite is smaller than host. And according to the place of parasite, we can divide them in two groups: ectoparasite and endoparasite. Ectoparasites are present on the body of the host. Endoparasites are present inside the body of the host. And sometimes some parasites can be both. For example, when there are some pathogenic fungi can be on skin as well as penetrating to cells. Okay, so those fungi are ectoparasite and endoparasite. Host specificity. What is this? Host specificity. Host, we know, is a living thing which one parasite attack. Let's see, one virus attack to human, don't attack to plant. One worm attack to a fish, not attack to human. So parasites are mostly specific, even though when one parasite come to our body, don't attack to whole of our body. For example, one virus attack to the nervous system, another virus attack to the intestine, another virus attack to the liver. So they have host specificity. Or one virus attack to E. coli cannot attack to the Pseudomonas. One virus attack to fish don't attack to a bacterium. So it is host specificity. Host specificity. It depends on the surface properties of the organism that allow physical attachment to the parasite to the host cells. So some cells are permissive, give permission to parasites to go inside and starting activity, and some cells surface is not permissive for another parasites. Very easily you can see when one, for example, if Plasmodium penetrate in our body, attack to liver and blood, not attacking to most other tissues. Or when hepatitis B virus come to our body, it attack to the liver. Or rabies, if come to our body, attack to the nervous system. So it is host specificity. To a specific host and to specific cells of host, they attack. One of the most important examples regarding to the parasitism is related to the viruses. Viruses. All viruses or most viruses are parasites. Why? See, some bacteria are parasites, some fungi are parasites, some protozoa are parasites. We use some for all microbes. All different microbes, some of them can be parasites, but regarding viruses, all of them are parasites. Why not some? Why all viruses? Because what is life? Life is metabolism, the sum of metabolism and anabolism. Where it happens in the cytoplasm. By help of what? By help of ribosomes present there and covered by cytoplasmic membrane. So for the simplest metabolism, nucleic acid, cytoplasm, ribosome, and cytoplasmic membrane all are needed. But viruses don't have ribosome, don't have cytoplasmic membrane, don't have cytoplasm. So they don't have their own metabolism. So viruses, for their lives, they are dependent on the metabolism of the other cells. So viruses must penetrate bacterial cell or human cell or plant cell to one specific cell, which allow it to a permissive cell and use the metabolism of the host. So accordingly, all viruses are parasites, and they are obligate intracellular parasites with great host specificity. Why viruses cannot live alone? Because they don't have tools, they don't have equipments necessary for metabolism. For active metabolism, they are dependent on other living cells, on those cells which they are permissive. So some viruses can infect bacteria, the others can infect human cells, the other can infect birds, and so on. There are viral parasites of bacteria, fungal, algal, protozoan populations. So we have different types of microbes as bacteria, fungi, algae, and protozoa, and all of these microbes have their specific viruses which can infect them. Parasitism may contribute to the disappearance of other fecal organisms in the sewage that enters aquatic habitats. Sometimes we can use viruses in biotechnology. For example, there are sewage from a city. It is full of organic matters and water, but the problems are pathogenic bacteria. So we can add bacteriophages or phages or those viruses which can infect bacteria. And after killing those bacteria, this liquid containing organic matter can transfer to the soil, to the river, to the sea. okay, before this primary treatment. And sometimes, for example, suppose one person is infected by one bacterium, and this bacterium is the causative agent for septicemia or sepsis. So there are a large number of bacteria present in the blood, and the person is very close to go to die, and those antibiotics are not working well. So in some special cases we use phage therapy. What do we do? In some cases we use phage therapy against sepsis. So as I told you, viruses are very host specific. So when we inject those bacterial viruses or bacteriophage to the body, they can only attack to the bacteria. They cannot attack to any cells of the body. So they can go there and they can kill the bacteria, pathogenic bacteria, and they can be positive for the life of the patient in some cases. Okay? So it was using the parasite against another parasite. So when we have septicemia, this bacterium is a pathogen, is a parasite in our body. So we use a specific parasite against another parasite. We call it phage therapy. Phage therapy. Sometimes when those sewage going to the river or going to the sea, fecal bacteria are in the sea and bacteriophage are there. In saline sediments, coliforms and E. coli and phage are absorbed onto clay particles, and E. coli are protected from phage attack, and E. coli can escape from the parasite. So as much as the salinity is more, bacteria can escape better from phage, and as much as salinity is low, infection of those bacteria by phages are easier. In lower salinities, they are absorbed from particles, and parasitic phage can attack and eliminate the host bacterial populations. Okay, the most important example which we explained was viruses. We want to go through another parasite. Bedelovibrio is a parasite. Bedelovibrio is a parasite, but it is not virus, it is bacteria. It is a type of bacterium, and this bacterium is parasite for another bacteria. Gram-negative bacteria can be infected by Bedelovibrio. Okay, so as you see in this figure, this is Bedelovibrio. Okay, this is Bedelovibrio, and it has monotrichus. It is equipped with one flagellum, and it runs very fast and attempting and trying to find a gram-negative bacterium a coliform like E. coli, Spirillum, or Klebsiella, or the others. With a simple contact, a permanent absorption attachment happens. So with a simple contact, never dissociation happens between parasite and host. And this host is a gram-negative bacterium. Then, by production of some enzymes and by pushing by the power of flagellum, it tries to go in the preplasmic space. When it goes, no need flagellum rotation, no need energy using for rotation of flagellum. So lose its flagellum and Bedello-Vibrio go to the periplasmic space. You know, whatever is down the cytoplasmic membrane is inside, and the other shells outside the cytoplasmic membrane are outside shells. So now bacterium is in outside shell of the bacterium, never go to the cytoplasm. It remains completely, for complete time, outside cytoplasmic membrane. It is present in preplasmic space, so it is ectoparasite. So it is ectoparasite and absorb nutrients in the cytoplasm as much as absorb food from cytoplasm, grows more and more and more till the time cytoplasm is empty and there is no food. And the appearance of host cell, as you see, for example, E. coli in this image, it is rod shape or bacillus. Host cell turns to a spherical shape and we call it bedeloplast. In bedeloplast, growing is ongoing till exhausted food, till limitation food. At this time, now cytokinase is starting and division, multiplication happens. Bedeloplast broken and the new Bedello-Vibrio are coming outside. They come outside. What are we talking about? We are talking about one bacterial parasite which attack to another gram-negative bacterium. Why do we talk about this? Because we are talking about interactions between two populations, two microbial population. One population is Bedello-Vibrio as a parasite, and the other population is the host. Host bacterium like E. coli is negative and harmed, and Bedello-Vibrio is positive plus and benefited, and it is benefited. So what did we told you? What did we tell you? Bedello-Vibrio, highly motile. Its speed 100 cell lengths per second. 100 cell length. For example, is your speed 100 of your length? Supposing you are 170 centimeter. Okay, or 1.70 meter. 100 times is 170 meter. Can you run 170 meter per second? No. So the speed of this bacterium is very fast than we are. Speeds 100 cell lengths per second and more fast than most other gram-negative bacteria. For example, E. coli goes 10 cells per second. Speed is 10 cells per second. After a small cell contact between parasite and host, result in permanent attachment of parasite to the host outer membrane. Then it goes to the periplasmic space, but not to the cell properly. Never go to the cytoplasm. Never penetrate the cytoplasmic membrane. So it is ectoparasite and loses its flagellum. For example, when I go home, I turn off the car. When you are in the destination, what is the need for energy using? The vehicle. So vehicle turn off in the destination. When parasite is in the periplasmic space, what is the need of energy use? For motility. 2. Host cell loses original shape and becomes spherical, so we call it pseudoplast. 3. Cell contents of host are partially degraded and utilized by predator, by the parasite with high efficiency. Why parasite is there? Parasite is there for partially degraded and utilizing those food in the body of the host. Number 4, Bedellovibrio grows into a filament without cell division. When host cell contents are exhausted, the filament divides into individual cells which develop flagella. Then bedeloplast lyses and releases the progeny, the new parasites. We have a definition with the name of burst size. We have the definition with the name of burst size. What is the burst size meaning? If one parasite go to host, how many new parasites are going out? It is burst size. What is burst size? If one parasite go to host, how many new parasites are formed and can go out? It is burst size. It is not a constant number. It is related to the size and quantity of food present in the host. As much as host is smaller, less food and less number of new parasites. As much as host is bigger, it contains more food and the result is more new parasites. For example, if host is E. coli, 4 new parasites, 4 new Bedellovibrio are formed. But if host is Spirillum serpens, 20 Bedellovibrio are formed. Why? Because Spirillum is bigger than E. coli, contains more food, so this filamentous shape of parasites are formed in longer shape and then divided and produce more Bedellovibrio. Is it clear? Can I go to the next for a few minutes? Maybe we can finish this part. Okay, thank you. So first parasite we explained was virus. Second parasite we explained was Bedellovibrio. And don't forget, we are teaching the interactions between microbial populations. Mostly we don't want to talk about pathogens of human. We want to talk about one microbe as a parasite attack to another population, microbial population, as the host. So in first example, viruses went inside the cell, the host cell. They were endoparasite. Bedalo vibrio, they were ectoparasite but attached, and they went to place of periplasmic space. In this example, we are talking about some parasites with the name of myxobacteria. Myxobacteria look like bedelovibrio, are ectoparasites, never going to the cytoplasm. But unlike bedelovibrio, don't attach as well to the host. They go very close. They go very close to the host, but never attach it. Never attach it. Myxobacteria is an ectoparasite, can cause lysis without direct contact. So they go to a close distance to host. They produce some enzymes, destroy cell wall and cytoplasmic membrane of host, and some leakage of nutrients is happening from these pores, from these destroyed places, and they start eating without contact. 1. Parasite produce exoenzyme that can lysis of susceptible microbial strains at some distances. Parasite derives its nutrition from material released by lysed microorganism. Soil myxobacteria are able to lyse gram-positive and gram-negative bacterial population. So they have more hosts than previous examples. They are effective on different gram-positive and gram-negative bacteria. We can have the same story like myxobacteria, but regarding cytophaga. Cytophaga has a similar story, but produce enzymes and lyses algae, photosynthetic algae. So myxobacteria is the parasite of gram-positive and gram-negative bacteria. Cytophaga is the parasite of algae. And any microbial populations which can produce chitinase, they can lyse fungal cell walls. Any microbe produce chitinase is the parasite for fungi. Some populations produce laminarinase and cellulase, and they can attack to the cell wall of algae and fungi respectively. So any microbe can produce these enzymes and lyses algae and fungi cell wall and cytoplasmic membrane. They are parasites of this populations. They are parasites of this populations. In all ecto cause lysis of host cells and use releasing nutrients, but they are not obligatory. For example, if myxobacteria cannot use one microbe going to another microbe, going to the other, so it is not any obligation. If they cannot receive food from this source, they go to another source. But some populations are resistant. Some hosts have some facilities for resistance to these pathogens, to these parasites. Some populations, some microbial populations can produce a spore and cyst. So when a parasite can easily receive food from a host, if it change its shape to spore or cyst, this relationship is stopped. So no parasite is present using spore or cyst as a host. Sometimes food is sufficient, pH is good, temperature is good, all the things are okay. But one microbial population change their appearance from vegetative form to spore form or cyst form for escaping the pressure of parasites. So sometimes you can see all the things are available for one bacterium or one protozoa, but they are not vegetative. They are in spore or cyst form. They change their appearance for escaping from pressure of parasites. Example 4, Legionella pneumophila is a opportunistic human pathogen. So if Legionella pneumophila cannot find a person with weak immune system, normally parasitize protozoa. Going inside protozoa, living, growing, and this protozoa as hosts are helpful for their survival and distribution in aquatic environment. Some fungal populations are parasites of protozoa, ecto and endo. So some hypha and mycelia are covered the protozoan and the others going inside the protozoa. So it is ecto and endo. Some fungal populations are parasites of algae, ecto and endo. Some fungal population parasitized by other fungal population. For example, we use mushroom in our pizza, and mushroom is related to Agaricus group of fungi. It is a type of fungi. But in this food industry, we have one problem with trichophyton, because trichophyton can grow on mushroom and make it not useful as using in food. So trichophyton is a problem. Why it is problem? Because trichophyton is the parasite of Agaricus as well as mushroom. And the last item, hyperparasitism. Hyperparasitism. Sometimes we have one parasite, like Bedelovibrio was parasite of E. coli. Bedelovibrio was parasite for E. coli. At the same time, phage is parasite for Bedelovibrio. So Bedelovibrio is a hyperparasite. What is the meaning of hyperparasite? When one parasite is a host for another parasite. For example, fungi is parasite of algae, and at the same time, this fungi is host for virus. So one parasite is host for another parasite. We call Bedelovibrio and this fungi as hyperparasite. One parasite serves as a host for another parasite. Last sentence, parasitism is a mechanism for population control. Parasitism results in the reduction of a host population density that then allows for accumulation and renewal of environmental resources being utilized by host. Okay, so it is what we should teach today. So don't forget, next time remind me to go to talk about the last interaction, predation. Predation is the last interaction between two populations. Okay, so next week, page 27. If there is any question, you can ask me. Okay, thank you very much. Hope the best. Have a good time. See you soon. Goodbye.